I'd like to thank the organizers for giving me the opportunity to present our work, which is on scoring transcript variation in single-cell RNA-seq data. Sequencing transcriptomes of single cells is getting more and more recognized, mainly because that it provides data at the cellular resolution. So first, it gives it shows the heterogeneity in gene expression levels for a particular gene in different cells. Say in this, sorry. Say in this toy example, I have 5 cells, and for this particular gene, we can see that it's expressed in very different levels in these 5 cells. And however, if we do bulk sequencing, we can only see the average expression level for this gene in these cells. Second, not only does the single-cell RNA-seq data show variation in gene expression levels, it also shows variation in isoform usage for some genes across single cells. So, so this is the region of this gene with 3 exons, and the middle exon is the alternative exon. We can see from the rich density profiles from the single cells that this exon is expressed in some cells and not in others. And again, with bulk RNA-seq data, we probably just see that it's expressed a little bit. So this phenomena has been reported in some recent publications. And traditional tools to study alternative splicing designed for bulk RNA-seq can be used to some extent with single cell data. For example, here, so the MESO, which can calculate exon inclusion scores, has been used in by Xiuwei et al. To show that there's high isoform variation in single cells. And they also show that the precise score, which is just the exon inclusion score, the precise score has bimodal distribution. And BAM2SSJ, which is an intron-centric method, has been used by Marinov et al. To study the number of isoforms for a particular gene in one single cell. And then, of course, mapping tools like TopHat and other tools which can find novel splice junctions have been also applied to single-cell data. And what we want to do is that we want to, basically, in the context of single cells, we want to do a global analysis of profile variation across single cells. So, for example, we ask questions, what are the sources of this variation? Which genes are significantly variable in isoform usage? And do these genes vary? Is there any pattern for these genes to use different isoforms, or is it completely stochastic? And also, with this information, with using different isoforms, does this information imply to us any subpopulations from a population of cells? To help us answer these questions, we first need a measure, basically, to quantify how variable are the isoforms used by a particular gene across a large number of single cells. So I start by introducing our profile variation score, which quantifies the isoform variation for one gene across a number of cells. And then, so this is basically going to be the basic tool used for later applications I will present. And in the second part, I will present some benchmarking tests that we have done, because in single-cell RNA-seq data, there is significant amount of technical and biological noise, and we want to see that our measure is reliable and is robust to this noise. And also, we want to have a reasonable threshold to say that at what point can we say that the isoform variation is significant and is beyond the technical and biological noise. And in the end, I will show a few applications where we try to answer the questions we had. Okay, so for the profile variation, which I will call PV hereafter, so I start with the read density profiles for one gene in different cells. So this plot shows this particular gene J, its reads are distributed in such a way in one cell and in a different way in another cell. So to quantify the difference between these two, or in practice we actually have a lot of these because we have a lot of single cells. What I do is that I normalize the read density profiles into probability distributions. So now I have probability distribution P1 and distribution P2. And what we do is that we calculate the Jensen-Shannon divergence between these probabilities, which is typically used to quantify differences among probability distributions. And formulate the Jensen-Shannon divergence of n probability distributions, which correspond to, in our case, n cells. So this divergence is defined as the entropy of average distribution minus the average entropy, where the H function is, of course, entropy, and the pi I is the weight for the ith distribution pi. And now, so the entropy H, we know that the entropy increases with the number of categories in a discrete probability distribution.
So this is like an information theory version of ANOVA or something, right?
Oh.
It's interesting. Yeah, never mind.
Okay, it might be. Except that I don't do a lot of statistics later. So the thing is that entropy increases the number of categories in a discrete probability distribution, which corresponds to, in our case, that corresponds to the gene length. If I don't do any binning, then my pi has the length of the gene length. And now, since I want to compare different genes, I want to compare genes of different lengths, so I don't want this bias. And because the growth factor of the entropy together with the length is roughly on the log scale, so we define our PV as the GSD divided the log of gene length. So this is so far we have a PV score, the profile variation score, and then we proceed to check how this profile variation score is correlated with other gene features. And the first thing we check is, again, so although we have normalized the gene length in the definition, we again want to check how it is correlated with the gene length after this normalization. So we see here there is still an obvious positive correlation between the PV and the gene length. This biologically makes sense because we know that for long genes do have higher potential to be alternatively spliced and to have higher variation. However, on the other hand, we also don't want this information to kind of become the dominant factor. We basically want to see what else is behind this gene length factor. So, and then we basically did linear regression to regress out the gene length factor, and the Y PV, the estimated PV the original PV minus the estimated PV, which is the residue, then becomes the another version of our PV score where the gene length is regressed out. So in the first part, basically, I introduced two versions of PV scores. One I call later original PV score, and the other the PV score with the gene length regressed out. And then we actually did quite extensive test to show that this score works. So I compared the PV scores from different data sets. And also, I also took different gene categories, including exons, and calculated the PV scores and see how different they are. And in particular, we for the thresholding part, we actually used the PV scores of exons, which I will give more details later. And also, we are also always interested to see if it is consistent with the results that we get from bulk RNA-seq data together with bulk RNA-seq tools, and then what is different. So various data sets. So here I took 4 data sets. The first 3 are all single cell data sets. This one has differentiating T helper cells, and then this one has embryonic stem cells from different culture conditions, and this one has embryonic stem cells from different cell cycle stages. The last one is interesting. It's a pool and split data set where all samples are supposed to be identical. So biologically, from how the cells were prepared, we know that the heterogeneity of these 3 data sets should be in a decreasing order, where the T helper cells are most heterogeneous, for example. So we basically see that the order of our PV scores also follow the order of heterogeneity in the biological setup. And what is interesting is that we see even for the pool and split sample, there is a range of variation which is basically caused by technical and biological noise. And then, so I said I also took genes from different gene categories and see how our PV works with different with these gene categories. So the first one, ERCCs, which has a distribution in purple. So these are the spikings, which are supposed to be very stable and conserved short sequences, and which are not alternatively spliced. So they should have the lowest PV, and which we see in this case is to the most leftmost. And then genes which are annotated with 1 transcript from ensemble annotations, their PV scores are shown in green, and then genes annotated with multiple transcripts have their PV values in blue. So for these 3 categories, we see that, okay, the order is also reasonable. So we are happy with this. And next, then we wonder, since we know that the PV score is contributed by technical and biological noise. So, say PV score and the PV score of actually alternative spliced gene would be contributed by technical noise, biological noise, and alternative splicing events. So how do we actually distinguish the alternative splicing events from all these noise terms? And so what we do is that here we actually make use of the exons. So the PV of exons is calculated over only the region of each exon. So I take the read distribution from each of this exon, and then across all these different cells, I calculate the profile variation for this exon. And then I do this for every other exon. So basically, the thought behind this is that the exons are subject to the same technical and biological noise. And the only difference between exons and alternative spliced genes in the same data set and the same experiment is basically the alternative splicing events. So if a gene has So basically, if a gene has significantly higher PV than the exon distribution, we can say with some confidence that its PV is contributed by the alternative splicing events. So this led us to plot the distribution of the profile variation scores of exons, which is shown in red here. And I'm just keeping the green and blue curve from the previous plot. And from the red distribution, which is the PV of exons, I take 5% of percentile, which basically gives me 5% of false discovery rate when I say that if a gene has its PV score higher than this value, if it's to the right of this point, then I say that its PV score is contributed by alternative splicing events. So this gives a way to extract genes which have significantly highly variable isoform usage. And with this criteria, we get around basically a few hundreds of genes found from each of the data sets that I've shown in a previous slide. And so then next we want to see what is in common of our single-cell data plus our single-cell method and bulk RNA-seq data plus bulk RNA-seq method. So the data I use here is a population of embryonic stem cells and a population of neural progenitor cells, NPC cells, which are differentiated from the ES cells. And so this is the single-cell data. With the single-cell data, I apply the PV score. And for the same cells, we also have we also sequenced the bulk RNA-seq. And so we have the bulk RNA-seq data for the ES cells and bulk data for the NPC cells. And then based on these two samples, I applied CAFDIF, which can be used to find differentially spliced genes between two bulk samples. So now the question is, what is in common between what we find and what CAFDIF find with the bulk RNA-seq data, and what else? What is extra thing that we find? So what is consistent? I'm just showing part of the results here. So where I divide all the genes into two categories. So one category is the genes which are detected by CAFDIF as differentially spliced, and the other set which is not detected by CAFDIF. And then I plot the PV of these two sets of genes. And so what we see basically is that the PV of the differentially spliced genes from the bulk RNA-seq data is higher than the other category. But then what are the genes which have high PV but not detected by CAFDIF? So because we have two cell types, and CAFDIF basically would be able to find those genes whose alternative splicing is caused by difference between two cell types. And then what we find but not found by CAFDIF should be the difference within one population. So what we actually see is that the genes which we found to have high PV but not detected by CAFDIF are enriched in cell cycle genes. So this makes sense because inside one population there's the cell cycle going on, and which is part of the biological variation within one cell type. So so now I can say that when I compare different data sets, the PV score conforms with biological heterogeneity, and also if I use the PV scores of exons, I can find significantly variable genes in isoform usage. And also when I compare with bulk data, there is consistency, and we also see what the bulk experiments cannot detect. And then I move on to a few applications of this PV score. Then the first one, which is obvious, is that we can find genes with high isoform variation, and then we can go on to ask what are these genes, what are they doing. And the second is then out of these genes, are there any 2 or more genes which change their isoforms in a correlated manner, or they just do things randomly. So here is that we try to find patterns in isoform usage. And the third one is if I have 2 cell types, can I find genes which have isoform switch from one cell type to another? So the first one, the first question, where we ask what are the genes with highly variable isoforms. When we did gene ontology enrichment, we actually see that the isoform variation comes from 2 levels, sort of 2 levels, which correspond to the 2 versions of profile variation score. So before we regress out the gene length, the genes which we see are enriched in functions like gene expression regulation and chromatin modification. So this is shared between different cell types. And after we regress out the gene length, we actually see more cell type specific functions. So for example, with T helper cells, we see a lot of immunology-related GO terms, and for ES cells, we see mostly the cell cycle GO terms. And so next question is, are there any 2 or multiple genes which change their isoforms in a correlated manner? So to answer this question, we first start with one gene. So for each gene, we first try to find its representative read-covered patterns. So because across, for example, 100 cells, maybe there are, say, 10 cells which share the same pattern, and then another 20 which share the same pattern. So I want to find all the representative patterns. When we to do this, we can easily use the square root of PV score. And again, here it shows the advantage of using this quantification is that the square root of PV, pairwise PV, is a metric, and we can perform hierarchical clustering on the matrix of square root PV scores. So for this one gene, we first get a PV score matrix, and then we perform hierarchical clustering, and we get a cluster dendrogram. And now basically we find optimal height to cut this dendrogram, and then we get groups of cells. And each group would correspond to one read-covered pattern for this gene. So now, here I'm just showing one gene and how the data looks like. So for this particular gene, called NSF, I found 3 representative patterns. And these, each color corresponds to one cell. And so basically, all the cells are grouped into 3 groups by these 3 patterns of read-covered profiles. So now basically I say that I can do this for every gene, and then if I have another gene, then another gene again by its representative patterns, it can divide the cells into different groups. If I have multiple genes which divide the cells into the same groups, then they are likely to be correlating in isoform usage. So I take multiple genes, and then I can make a matrix. I can construct a matrix like this. So for gene 1, if it uses pattern A in cell 1, then I write it here. And say, for example, if it uses pattern B in cell 4, I write pattern B here. And blank cells are here correspond to the cases where the gene is actually not expressed in this cell. Just note that the pattern A and pattern B for each different gene, so this pattern A doesn't equal to this pattern A. I'm using AB just to distinguish different patterns. So now it looks like something which we can probably do clustering or some similarity measure, which and to find correlating columns. I have to say that the difficulty of this question is really because that there are a lot of blank cells. So this matrix in real data is actually very sparse. Yeah, 5 minutes. Okay. What we do then, I basically expand. So for each gene, I expand its columns by the number of patterns it has. So now I have gene 1 pattern A, gene 1 pattern B, and then gene 2 pattern A, and then gene 2 pattern B, etc. So if it uses pattern A in cell 1, I have a 1 here, and then otherwise I have 0. And these red zeros correspond to the case that this gene is just not expressed in this cell. And now I have a binary matrix which we can easily work on. And a simple thing that we tried is just to calculate the Jacquard distance between all the columns. And then we can, again, build a dendrogram tree based on the Jacquard distance. And with if we choose a particular threshold to cut this tree, we can see what subtrees we get, which would correspond to a set of genes. So from this data, we actually see that we don't. So with this particular threshold, we actually don't have a lot of subtrees produced. So it looks basically in general like this. The whole pattern is very stochastic. So we also compared with random binary matrices where basically I shuffled this original matrix, and we see that the real data is less.
Can you do the same for the exons, and that would give an idea of where the reliable line to cut?
You mean how do we find the threshold?
This is for the introns, right?
This is for the whole gene.
Okay. Because the question is, how do the same control as you did for the P value? Maybe you can do a similar control here, and that will tell you where you kind of go beyond just stochasticity in the data.
Yeah. That's we actually did something similar. So basically, I varied this height. I basically moved this level from bottom to top, and then at every step I compare it with the randomly generated binary matrices. So what we see is that.
But they kind of don't know. Random binary matrices don't know about stochasticity in data.
So random basically gives a control of what completely random is. So because when we look at the data, we see it looks random, but if it is just theoretically completely random, or if it just looks random and we just didn't find the pattern. So the reason that we generated random binary matrices is basically to have control for that.
But the biological question: are these really random isoforms? Or is it noise in the single cell data?
Yes. So the biological question is that. So I don't have a final conclusion to answer that question. So basically what we know here is that the data that we see gives us a very small number of these correlated genes. And so this is also partly, as I said, due to the stochasticity in gene expression itself. So if a gene is just not expressed, then we don't have the information for that. So now I basically say that maybe I'm asking for too much if I ask for genes which change their isoforms in a correlated measure. Now I'm going to change my problem a bit. So I'm saying that, okay, I don't ask you to find the cell groups. Now I'm basically telling you, so these are two groups of cells. And I'm also not looking for multiple genes. Now I'm just looking at each single gene and see if this gene uses different isoforms in different cell types. So this becomes my the last application that I want to show. So how do we, is that with this PV measure, we can find genes which switch isoforms between cell types. So again, I'm using this ES cell and NPC cell data set. And now basically for each gene, I also construct a square PV distance matrix. And for all the ES cells, I call all these distances intergroup distances. If it's between ES cell and NPC cell, then I call it intergroup distances. So basically, the reasoning is that we want to find genes which have a high ratio of intergroup distances over intergroup distances. And also, of course, we want it to be from this. We want the gene to be from the set with significantly high PV, which we identified earlier. So with this method, we found a set of genes, and I'm just showing one example of them, which actually looks very interesting. So this Lig1 gene, so we found two representative read density profiles. One is a long isoform, and the other is a very short very short transcript. And then basically these are the cells, and again each cell, each color corresponds to a cell. And these are also another group of cells. What we see is that in this group of cells, we actually have mostly ES cells and a small number of NPC cells. And in this one, we have all NPC cells. So is this, and do they have, another question that we ask is, do they have protein products, which might actually lead us to some functional conclusion? So I go to check the protein domains. Now for the long transcript, it has 3 important domains, and then for the short one, which we see that this is only like to TL 330, which corresponds to only this point here. So the short transcript basically lost all of this, and it's kept only this. So it looks like there's changing function too. And then we further look at check what this gene is doing. So then we found that Lig1 is actually a cell cycle gene. And this is combined with the fact we know that when cells differentiate from ES to NPC, they actually slow down significantly the cycling. And so basically, based on this particular gene, what we see is that from ES cell to NPC cells, this gene Lig1 is actually changing its isoform usage, but however in NPC cells it's still keeping some NPC cells is still keeping the original isoform, probably just because it's delayed or probably just because it's stochastic. But this is what we can find with single cell data. And I need to say that this particular gene is not detected by CAFDIC with the single cell data. So this also shows the advantage of using the single cell data. Okay, so by this, I want to conclude the last part where with the PV score that we designed earlier, we can explain partly the sources of isoform variation, and we found very high stochasticity in isoform usage across the whole data set. And we can also find genes which switch isoforms from cell type from one cell type to another, which very likely correspond to its function change during differentiation. And I want to thank my postdoc supervisor Sarah Teichmann and Valentin Jong Kim, who I had very useful discussions, and Oli, John, Nick, Kaur, whose suggestions have also been very useful. And I want to thank Swiss National Science Foundation for offering me a fellowship to support my postdoc research. Thank you.
So how many cells in the bottom panel, the second splicing form, how many actual cells were there, the neural precursor cells only? In that
you mean,
3 slides back, you said like 7 and 12 or something, or 5 and 12? And then
so,
how many over there?
I mean, here there probably you can see probably like 15 or something, or 12. But in the data set we have, say, a few chips that would be like maybe 300 to 400. The thing is that again, in a lot of cells, this gene is not like for a lot of genes they are not expressed in a lot of cells. So I took only, so these are all the cells which express this gene.
I was just wondering where you could compute some kind of hypergeometric p-value or something.
Not in this case. So yeah, I tried to basically back this up with like other evidence.
What are these red and blue bars?
Yeah, sorry. So these are just the exons. So
you take out intron when you do this.
Yeah. So for this one, for visualization, I'm taking out the introns. Yeah. So this.
When you calculate PV, do you also take out intron?
Yeah.
So how do you account for like intron retention?
Right. So in this case, we are actually not using the introns. We do see expression on the introns, but so basically because we don't know what they are, so we decided just to basically remove the introns and for the PV calculation.
Thank you so much.
Thank you.